Hello students, welcome to classroom. This is our new YouTube channel. This is Moumita ma'am, and we would discuss the board topics of biology one by one. Today we would discuss cell cycle and chromosome structure, the very first chapter of your board syllabus. At first we would discuss the topic cell cycle, and then we would discuss the chromosome structure. So what is cell cycle? We, the living being, we born, and then we grow, and then we reproduce and give rise to our progenies. Just like this, the unit of life or unit of the living beings, the cell, they also get born, and then they grow, and then they give rise to their new progeny cells. And this whole thing is known as the cell cycle. This occurs in a cyclic way. As they give born to the new cells, these cells also then grow, and then they also produce new cells. So this is a cyclic thing, and this is and that's why this is called the cell cycle. So just like the life cycle of any living being, cell cycle can be divided into two phases. First is interphase, and then the mitotic phase. Just like us, as we first grow and then we reproduce, the cell also first grow and then it reproduce or divide. The first phase or the interphase is also known as the growth phase, because this is the phase of growth. This interphase thing is also then divided into three stages. G1, A's and G2. There was not enough space in the board, that's why we changed the board. We just only discuss the interphase topic in this board and then the mitotic phase. So see that interphase is divided into three stages, G1, S and G2. Here the G signifies the word growth, right? Now in G1, what happens in G1? As we first grow, first born, and then we grow in size, right? So the cell is also grow in size in G1. How they grow in size? They metabolize and they produce all the different RNAs and the other proteins for the growth and development of the cell. Okay, so in G1 they grow in size, metabolism, occurs in its peak, synthesis of RNAs and proteins. These things occurs, or these things happens in G1 phase or the growth 1 phase. After that, the phase is named as the S phase. This S means synthesis. S means synthesis. What things synthesized in this phase? The most important thing of the cell. What is that? That is chromatin or the DNAs. Okay. So in synthesis phase or S phase, DNAs are synthesized in this phase. This is a phase of DNA synthesis. So we can see that the synthesis stage is the synthesis of DNA molecule. here in this stage the 2N DNA molecule become 4N, as it makes one of its copies. Each of the DNA molecule makes its another copy, and they become 4N. Okay, so this is the most important portion of interphase, and we would get the result in the mitotic phase. When we discuss the mitotic phase, we will discuss this thing also. Okay, the next phase of interphase is G2, or the growth 2 phase. This is also called as the pre-mitotic phase, and in this phase the proteins which would be required for the mitotic phase are get synthesized. So this is also a stage where proteins for mitosis get synthesized, and the mRNAs or the messenger RNAs, mRNA or the messenger RNAs, and all the tRNAs or the transfer RNAs get synthesized in this phase. Okay, so we have discussed all the 3 stages of interphase: G1 phase, S phase, and the G2 phase. But sometimes some cells don't go through all through 3 of these stages of cell cycle of the interphase. These cells are said to be in a G0 phase, G0. Here G don't signify the word growth. Here G means gap. Those cells which don't follow the stages of interphase of the stages of cell cycle, we call them to be in a G0 phase or in a gap phase. Which cells are these cells? The cells, those don't divide or don't divide any time, just like our neuron. As we know that neuron so neuron is always stay in a G0 phase, as neuron doesn't have any centriole. That's why neuron can't divide into another neuron. That's why we say that neuron is always in a G0 stage. And we can discuss about the RBC, as RBC doesn't have any nucleus. That's why RBC also can't divide and produce another RBC. That's why we can say that RBC is also in a G0 phase or a gap phase. Okay, so we and another kind of cells we should discuss in this G0 topic, that's our cancer cells. As we know that cancer cells don't follow any part or any rule of the normal cell cycle of the interphase, and they just get divided and divided, and that's why we say that cancer cells are also in the G0 cell, or they stay in a G0 or gap phase. Okay, so those cells which doesn't follow the rules of the cell cycle or the normal stages of cell cycle, we call them to be or to stay in a G0 stage. So when the cell completes all the stages of interphase properly, it gets permission to go in the mitotic phase or to divide itself in the progeny cells. Okay, so we will discuss now the mitotic phase. The mitotic phase is being divided into 2 main parts, which are known as karyokinesis and cytokinesis. So here karyokinesis, at first the karyokinesis, and we will discuss then the cytokinesis. So here the word kinesis means the division and karyo means nucleus. Okay, and on the other hand, kinesis, as we discussed earlier, that kinesis means the division, and cyto signifies the cytoplasm. So in this process, at first the nucleus would divide and then the cytoplasm divides. Okay, so the nucleus division or the karyokinesis is then can be divided in 4 main phases. We will discuss these 4 main phases. but not in this board because there is not enough space. We will discuss the stages of karyokinesis. The karyokinesis can be divided into 4 stages: prophase, metaphase, anaphase and telophase. At first we will discuss the first one, the first stage of karyokinesis, which is known as prophase. Here the name pro means the very first stage. This is the very first stage and this is the longest part or the longest stage of karyokinesis. This stage or this phase could be further divided into 2 stages: the early prophase and the late prophase. In the early prophase, we can see that the nucleolus and the nuclear membrane is intact in the cell, as we have drawn on the board, and the chromatin is also intact. But in this phase, the chromatin starts to get condensed and form chromosomes. When chromatin attached with the other protein parts and it get condensed, it makes some definite structures which are known as chromosomes. Okay, so in the earlier stage, when the prophase just begins, we cannot see the chromosomes in the cell. But when it moved to its later stage, the prophase moved to its later stage, we can find the permanent and the perfect structures of chromosomes in the cell. And as we drawn on the board, that the nucleomembrane and the nucleolus also degenerates in this later stage. Okay, so the first 2 points we can write on the prophase, that the chromosomes. So as we can see that the first and the most important 2 properties of prophase are that the chromatin condensed and form the chromosomes, and the nuclear membrane and the nucleus get degenerated. Okay, and in case of animal cells, as we know that animal cells have a special type of organelle which is known as centriole, which the plant cell doesn't have. The centriole gets separated from each other, and both the centriole goes to the different pole or the 2 different pole of the cells. Okay, this movement starts in the late prophase stage. So we can see that these are the 3 main points: that chromatin condensed and forms the chromosomes, nuclear membrane and the nucleolus gets degenerated, and centriole divides and move towards the opposite sides of the cell. Okay, this thing occurs only in animal cell, not in plant cell. Okay, now we will discuss the topic, the second part or the second phase of karyokinesis, which is known as the metaphase. This is very important and most important, I should say, in all the 4 stages of the karyokinesis. In metaphase, we can see that the chromosomes now get arranged in the middle portion or the equator portion of the cells. Here the chromosomes get arranged in the equator portion of the cells, and they get attached to some thread-like structure we can see on the board. These thread-like structures are known as the astral rays in case of the animal cells, and in case of plant cell we call it the spindle fiber. Why the 2 different names are here? Because in case of animal cells, these thread-like structures are made from the centriole. As we discussed earlier in prophase, that the centriole get divided and move towards the opposite poles of the cell. In metaphase, this movement completed, and these fibers or the threads generated from the centriole from the opposite poles, and these threads are get attached to the chromosomes in the equator portion. Okay, and in case of plant cells, as there is no centriole, these thread-like structure occurs with the help of the microfibers or the microtubules which are present in the cells. So these 2 different names we have given to them. In case of animal cells, as they looks like the astral structure or like stars, that's why they are known as the astral rays. And in case of And in case of plant cells, it is known as the spindle fibers. So we can see that the chromosomes are arranged in the equator and the chromosomes are attached to the astral ray in case of animal cell, and then fibers in case of plant cells. Okay. Now we have to discuss some more things in this phase, that the chromosomes are now see, you can see that this is not the structure of a regular chromosome. Here the chromosomes are divided into the chromatids. Okay, so what is chromatid? As we have discussed earlier in the interphase stage, the A stage of interphase, that the DNA produces its own copies, and the 2n DNA become 4n in this in those stage. Remember, so that in that A phase, DNA make their copies. These copies get now detached in this phase, the metaphase, and they form this kind of structure where they only get attached with the centromere region, but the two arms of the chromosomes get detached from each other, and these things and in this stage we can say them not as chromosome, we should say them as chromatids, and these chromatids are attached. So we can see that the chromosomes are get attached, or the chromatids are get attached with the threads. These threads are of two types. Some threads are attached with the chromosomes. These threads are not continuous threads. These are known as discontinuous threads. And the other threads which I have drawn with a different color, yellow, these threads are not attached with the chromosome, and we say them as a continuous threads. Okay. So these are the main important characteristics of metaphase. After this stage, the cell or the cell cycle or the karyokinesis move to its the next stage, which is known as the anaphase. Now we will discuss the third phase or the third portion of the cell cycle of the karyokinesis, which is known as anaphase. In this phase, the spindle fibers get shortened, and as a shortening of the spindle fibers, because of the shortening of the spindle fibers, a pull generates at the centromere region of the chromatids, and both the chromatids get separated from the centromere, and both the set of the chromatids started to move towards the pole of the cell, and this movement is known as the anaphysial movement. So this is the shortest portion or the shortest phase of karyokinesis. Now we have reached to the last portion or the last phase of the karyokinesis, which is known as the telophase. Here, tail means the last portion. Tail, T-E-L, tail means the last portion in genetics. Okay. Now we can see that both the set of the chromatids, which now are not chromatids, they are the daughter chromosomes or the new chromosomes, they have reached to the pole, as we can see from the board, and the nuclear membrane and the nucleolus have reappeared in these stage, as we can see here. And now we can see that there is a single cell where two daughter nucleus are appeared. Right? So this is just like the opposite of prophase, where the condensation occurs and the chromatids becomes the chromosomes. Here the decondensation occurs and the chromosome now again will be the chromatid. Okay, and the reticulate structure of nucleus would reappear. Okay, so we can say that the characteristics of telophases are nuclear membrane and nucleolus reappears, two daughter nucleus appears, as we can see from this drawing. Okay. So here the karyokinesis ends, and after this completion of karyokinesis, we would study the cytokinesis. So we have completed the topic karyokinesis or the division of nucleus. Right now we will study the next portion of cell cycle, which is known as the cytokinesis. This is the second part of the cell division or the mitotic phase. Here the cytoplasm divides and thus the cell divide into the two daughter cells. As we can see from the pictures, this occurs differently in animals and plant cell. At first we will discuss the animal cell and then we will discuss the topic in the plant cells. Inside animal cells, the cytokinesis occurs in a very simple manner. At first, when the two nuclei or the two daughter nuclei appears on the cell, we can say, as we can see from the picture, here a furrow appears in the equator region, a furrow. This furrow, after appearance, it gets deepens and forms a cleavage like structure, and then both the cleavaged cell membrane get attached to each other, and thus the cytoplasm gets separated from each other and the cell membrane gets separated from each other, and the two and now we can see as I am drawing here, and then the two daughter cell appears. Okay, this is the process of cytokinesis in the animal cell. Now we will discuss the cytokinesis in plant cells. In case of plant cell, the cytokinesis actually begins with the appearance of the interzonal fibers. These interzonal fibers appears during the anaphase, the late anaphase stage. And after appearing these interzonal fibers, it gets condensed, and after that, when the two nuclei appears completely, these interzonal fibers get fused with the Golgi bodies of the cell. All the Golgi bodies in the plant cells get fused with this interzonal membrane, interzonal fibers, and both of them after fusion they make a plate-like structure which is known as a cell plate. This cell plate gradually develops and forms the middle layer of the cell wall. As we know that the plant cell wall can be divided in three layers: the middle layers, and after that the primary layer, and after that the secondary layer. This middle layer actually appears at first, which is formed from the cell plate which appears during the cytokinesis. So the cytokinesis in plant cells occurs with the help of the cell plate. So we have studied all the cytokinesis in animal cells and the plant cells. As we can see from the picture that in case of animal cells, after the division or the cytokinesis, both the daughter cells get separated from each other. But in case of plant cells, as we can see that they don't separate from each other. They just cell wall appears in between the daughter cells, and thus the cytokinesis or the cell division completed. As the cell division completed, we get some two new daughter cells in case of animal cells or in case of plant cells, and as we get daughter cells, the new cell cycle or the new cycle for these new cells started. So this was the first topic, cell cycle.